Epithelia form the surface layer of the body, line body cavities, hollow organs, and structures, and constitute most gland tissue. Any substance that enters or leaves the body must cross an epithelium. All epithelia consist of one or more layers of tightly packed cells and little or no extracellular matrix between cells. Distinguishing features of an epithelium include a free apical surface and a fixed basal surface. Specialized intercellular junctions join adjacent cells, and a thin basement membrane separates the basal surface from underlying connective tissue layers. Although epithelia lack blood vessels, many sensory nerve endings are present. Epithelia have a high regenerative capacity that allows them to maintain their function. The primary functions of an epithelium are to resist dehydration and injury from physical, chemical, and biological agents, to selectively regulate materials entering and leaving the body, to secrete products produced by epithelial glands, and to monitor the environment through specialized sensory nerve endings and specialized epithelial cells. Epithelial tissues are classified using two basic criteria: the number of cell layers and the shape of the cell at the apical surface. An epithelium composed of one layer of cells is termed simple. If there are two or more layers of cells, the epithelium is termed stratified. When the apical cells are flat and thin, the epithelium is classified as squamous. Apical cells that are somewhat square-shaped belong to a cuboidal classification, while those with a rectangular shape are termed columnar. By combining the criteria of cell shape and number of layers, all epithelia can be classified easily. For example, a simple squamous epithelium is composed of a single layer of thin, flat cells. In contrast, a stratified squamous epithelium is composed of multiple layers of cells, with the apical surface layer being thin and flat. These same tenets could be applied to cuboidal and columnar epithelia. There are two special types of epithelia. Transitional epithelium, found in the urinary tract, is a stratified epithelium that gives the impression of transitioning from cuboidal, when the epithelium is relaxed, to squamous, when it is stretched. A pseudostratified epithelium, found in the trachea, for example, is a simple epithelium in which all cells contact the basal lamina, but only some reach the apical surface. Cells are of varying heights, and their nuclei are at different positions in the epithelium, giving the impression of stratification. Some epithelial classes have specializations of the free surface of apical cells. Simple columnar epithelial cells in some organs have cilia, which are small, finger-like motile projections. Thus, simple columnar epithelium is subclassified as ciliated or non-ciliated. In parts of the digestive and urinary tracts, epithelial cells involved in absorption have a dense collection of short cytoplasmic processes called microvilli on their free surface. Microvilli are non-motile and serve to increase surface area. Because the microvilli appear fuzzy in the light microscope, cells with numerous microvilli are said to have a brush border. A stratified squamous epithelium has either dead or living cells as the surface layer. The dead cells contain the protein keratin. Therefore, stratified squamous epithelium is subclassified as keratinized or non-keratinized. Epithelial cells are tightly connected to adjacent cells by specializations called intercellular junctions. There are four types of junctions: tight junctions, adhering junctions, desmosomes, and gap junctions. Tight junctions, also known as zonula occludens, are bands of fused cell membrane near the free surface of adjacent epithelial cells. These junctions attach a cell to its neighbors, encircling cells and sealing the space between them. Thus, substances crossing the epithelium must pass through epithelial cells, not between them. Tight junctions are essential to the primary function of an epithelium, which is to regulate substances that enter and leave the body. Adhering junctions, also known as zonula adherens, have a similar band-like arrangement seen in tight junctions. They are located deep to the tight junctions and have numerous microfilament proteins from the cytoplasm that act as anchors for adjacent cells. There is a small gap between the cell membranes of adjacent cells at adhering junctions. Desmosomes, also known as macula adherens, are small button or snap-like junctions. These form at the points of mechanical stress. They are different from tight and adhering junctions in that they do not encircle the entire cell. Gap junctions are points where small pores connect the cytoplasm of adjacent cells. The gap junction consists of a series of hollow bridging structures called connexons. Small solutes, such as ions, glucose, and amino acids, pass through connexons from the cytoplasm of one cell into a neighboring cell.